Doxycycline is an antibiotic used for treating different conditions including acne, chlamydia, and syphilis. It comes in many forms like tablets, capsules, or injection. But how did scientists find out that this could be used for treating infection? To understand this, we need to take a trip back in time and look at the history and development of doxycycline. It was all started in the 1940s. Benjamin Duga collected soil samples from all over the world. After analyzing them, his attention was drawn to an unknown compound that proved to have impressive antibiotic activity. The new substance was isolated in 1945, and due to its unusual yellow-colored colonies, it was called chlorotetracycline. Meanwhile, scientists from Pfizer independently obtained soil samples and also isolated a new compound called oxytetracycline. The chemical structures of both compounds, however, remained a mystery until 1952, when scientists from Pfizer finally discovered the structures of both drugs. More modifications were made to produce a more active analog called tetracycline. By mid-1950s, there were then 3 new drugs used clinically: chlorotetracycline, tetracycline, and oxytetracycline, forming a new class of drugs, the tetracyclines. After seeing the success the modifying molecules can bring, Pfizer chemists chose to modify the C-ring of oxytetracycline to afford chemical stability. Halogenation and dehydration of the C6 gave rise to the new antibiotic methicillin. Although it wasn't approved for clinical use, it served as the starting material for another Pfizer scientist, Charlie Stevens. He realized the new chemistry had to be developed to C6. The reduction of the double bond gave a compound that showed remarkable pharmacological activity. The molecule had better stability and increased lipophilicity due to the absence of the OH group at C6, which facilitated its absorption. This new compound was then called doxycycline. Now that we know how the molecule looks like, we need to determine what makes it active. Let's start by looking at the structure in more depth. Doxycycline is made up of a DCBA tetracene ring system, and it contains important functional groups such as an alcohol, a phenol ring, a ketone, an amide, and an amine. Before we dive into the chemistry of this molecule, let's explore whether this molecule obeys Lipinski's rule of 5. For this molecule, the molecular weight is 444.4, and it has a log P of -0.2. Hydrogen bond donors and acceptors also affect the oral availability. How many do you think there are? There are 6 hydrogen bond donors and 9 hydrogen bond acceptors. Therefore, it doesn't fit the rule. Despite this, doxycycline is orally available. An active tetracycline with antibacterial activity, such as our drug, must have a pharmacophore consisting of a straight DCBA tetracene ring system, an amide group at C2, amino group at position C4, which must be in the S isomeric form, hydroxyl groups at C3, C10, and C12, as well as carbonyls at C1 and C11. The D-ring is the most flexible one to change. All modifications carried out in this region of the molecule would make the drug more specific to a bacterium. None of the chiral centers, except C6, should be modified as it leads to detrimental loss of activity. Next, we will look at the synthesis of doxycycline. The entire process involves 18 steps, and it was developed by Andrew Myers and his colleagues at Harvard University in 2005. The starting material is benzoic acid, and it undergoes microbial dihydroxylation in a reaction catalyzed by the bacterium A. eutrophus. The next steps include an epoxidation reaction which occurs readily, as well as the addition of a couple of protecting tert-butyl-dimethyl silyl groups. This produces an enantiomelically pure ester. The resulting molecule then undergoes an acylation reaction with dimethylamino-substituted isocosol, forming a ketone. After exposure to the enzyme lithium triflate, the molecule undergoes a selective desylation reaction with trifluoroacetic acid, which removes the TBS protecting group. It also opens up the epoxide, which is thought to be an SN2 reaction, forming the alcohol as shown here. The next step includes a nucleophilic substitution reaction using thiophenol, where a hydroxyl group is removed. Then trimethylphosphite, a desulfurization agent, removes the thiophene group from here. Carbon 5 becomes oxygenated in this step, and an enone is formed here. In the penultimate step, a cyclization reaction occurs by conjugate addition of a phenyl ester to the enone formed in the previous step. In the final step, a deprotection reaction occurs where the protecting groups, TBS and BOC, are removed by reacting with hydrofluoric acid. This produces doxycycline. Overall, this is a highly efficient and diastereoselective synthesis. However, the way in which doxycycline is commercially produced is by a semi-synthesis reaction developed by Pfizer chemists. Semi-synthesis usually involves using chemical compounds derived from natural products as a starting material, so in this case, methylcycline is that starting compound. So this process involves the reduction of the double bond at the C6 in methylcycline, which is a stereospecific reaction catalyzed by rhodium carborane complexes, and doxycycline is produced in this one-step reaction. The semi-synthesis is preferred to the total 18-step synthesis as it is a cheaper option with fewer chemical steps. Doxycycline is very useful when it comes to treating acne. Let's see what causes acne and how our drug can help. Acne is a common problem found in most teenagers once they hit puberty. It is a chronic skin disease involving the blocking and inflammation of pilosebaceous units. This affects mainly the face, but can also be found within other regions of the body, for example, the back or chest. This is because it typically affects areas of skin with the densest population of sebaceous follicles. Local symptoms can include pain, tenderness, or redness of the skin. The main established cause of acne is genetics. Acne develops as a result of interplay between four main factors: follicular hyperkeratinization, followed by plugging of the follicle, excess sebum production, the presence and activity of commensal bacteria, and release of inflammatory mediators in the skin. The severity of acne can, however, be broken down into four categories: comedone, mild, moderate, and severe. Therefore, in relation to doxycycline, it is used for moderate to severe acne. It works against the protein synthesis of Propionibacterium acnes, which then prohibits the development of acne. P. acnes is an anaerobic gram-positive bacterium present in acne lesions. It stimulates inflammation by producing pro-inflammatory mediators that diffuse through the follicular wall. P. acnes activates the toll-like receptor 2 on the monocytes and neutrophils. This then leads to the production of multiple pro-inflammatory cytokines. Hypersensitivity to P. acnes explains why some sufferers develop inflammatory acne vulgaris while others don't. So now that we've discussed what doxycycline is indicated for, let's discuss how it works and look at its mechanism of action. Most commonly used antibiotics are bactericidal and work by breaking down the cell wall. However, doxycycline is bacteriostatic and inhibits protein synthesis. The tetracyclines are broad-spectrum antibiotics with activity against both gram-positive and gram-negative bacteria. In gram-negative bacteria, the tetracyclines cross the outer membrane by passive diffusion through porins and pass across the inner membrane, which is dependent on a pH gradient. As we've mentioned, acne is caused by gram-positive bacteria. Doxycycline works in the treatment of acne by inhibiting translation and therefore inhibiting protein synthesis in the gram-positive bacteria. This happens in the ribosomes. In the absence of doxycycline, the charged tRNA will bind to the A side of the ribosome and then transfer the amino acid onto the new peptide chain. However, when doxycycline is present, it binds to the 30S unit of the ribosome, inhibiting tRNA from binding to the A site. Therefore, the amino acid cannot be added to the peptide chain, leading to the inhibition of protein synthesis. When doxycycline comes into contact with the ribosome, it gets involved in an extensive network of hydrogen bonding interactions with the phosphate backbone of the ribosomal RNA. A cytosine ring is also involved in pi-pi stacking interaction with the D ring. Following the mechanism of action, like many drugs, doxycycline also causes some side effects, including dry mouth, fungal superinfection, and diarrhea. Fungal superinfection is when mutations in the RNA can affect the binding site and weaken the binding interactions, therefore creating a resistance against tetracyclines. This leads to the formation of another infection. Antibiotics also usually cause diarrhea. The reason why this happens is because when the drug is targeting the 70S ribosome of the bacteria, it ends up getting rid of the bacteria that is useful for digestion, causing an upset stomach, leading to diarrhea. This drug also has some contraindications, just like most of the drugs on the market. One of the biggest contraindications is alcohol dependence. As alcohol has an enzyme-inducing effect, it mainly affects drug metabolism. This means the doxycycline will be metabolized quicker, thus reducing serum levels of the drug. The other main contraindication is children under 12 years of age, as doxycycline gets deposited in growing bone and teeth by binding to calcium, causing staining and occasionally dental hypoplasia. There are some important interactions with other drugs to notice. The main drug interaction is between doxycycline and statins, for example simvastatin. Antibiotics generally have an effect on the liver, affecting the cytochrome P450 enzyme. This enzyme metabolizes toxic compounds such as drugs. In presence of doxycycline, this enzyme is inhibited, therefore statins are not metabolized. This leads to chemically induced liver damage called hepatotoxicity. In conclusion, doxycycline is a very useful antibiotic from the tetracycline family. It is commonly prescribed and dispensed in community pharmacies for many different conditions. Thank you. We hope you enjoyed this video produced by Group 11 with the contributions of Lucy, Sharon, Adnan, Daniela, Alvin, Will and Sandra.